All right, so now I can share my screen, and I'm going to start the lecture for Sophie and Burke. And just let me know when you want me to scroll to the next slide.
Okay, so my name is Sophie Chu. I am a research scientist at the University of Washington and the NOAA Pacific Marine Environmental Lab. Burke is a professor at Oregon State University. I'm going to go over some background on the CO2 system and before we start going into different monitoring portion, the monitoring portion of the presentation. Okay, next slide, please.
All right, there we go.
So some background on, in general, like the global carbon cycle. We have all these different reservoirs of carbon in the rocks, in the ocean, and in the atmosphere, in the land, in the ocean, and the atmosphere. And so here in this slide, you can see different fluxes from each of these reservoirs. And the black fluxes are natural, and the red ones are anthropogenic, or from human activities. And so you can see, you know, back and forth fluxes. In the ocean, you have some input from rivers. You have interactions between the ocean and the atmosphere. And in general, what I wanted you to get from this slide is that there is a carbon dioxide CO2 uptake for the ocean that is about 27% of the total anthropogenic CO2 emissions. Next slide, please.
Sorry, it's toggling between programs. There you go.
Can you play? There's both of these are kind of short, sort of. There you go.
Yeah,
yeah,
I can play it.
That show kind of how the carbon has accumulated in these different reservoirs over time. And so and that one just showed, in black you have the fossil fuel and cement production, which is a big source of carbon.
Oops, sorry, it just scrolled ahead.
No worries. And then you have atmosphere taking up some of that CO2 in red, and then also land in green and ocean in blue. And you can see how each of those reservoirs have taken up carbon. And then in the right side, this is another movie that will show you. So on the left side, you have the sources from fossil fuels and land. So land is mostly just a residual. So basically that means we can estimate pretty well how much fossil fuel contribution to increase in CO2 is. And we can estimate how much the land and atmosphere are taking up. So those are what the sinks are on the right side. And then the residual amount is just what we estimate from the land, because that estimation is just has a large uncertainty.
It looks like we are getting a lot more people joining. This is great. So we just started the presentation. We waited a little bit. So don't worry. And we are recording this, so if there's anything you missed, you'll be able to go back and look at it.
So I think this on the right side shows you pretty well how much of the increase in...
Sorry, it's my mouse has a mind of its own today.
Okay. No worries. Yeah, so you can see how much of those emissions are being taken up by the atmosphere and ocean over time. we can wait until it finishes and then move on. Okay. Okay, so that that increase in atmospheric CO2 is plotted. So first I'm going to talk about the left side, is plotted on in the red, and it has led to a direct increase in a direct increase in partial pressure of CO2, PCO2, in the ocean. And then that has led to a decrease in pH, which is what we call ocean acidification. And then on the right side is just showing that same CO2 increase, but with a bit further along in time, so the last 10 years included. And I pulled the CO2 concentration in the atmosphere from yesterday, and that's at 413, which is, you know, very high in historical CO2 record. Okay, next slide. Okay, so now we're going to talk about what happens to that CO2 that comes in from the atmosphere when it gets absorbed by the ocean. So there's 4 carbonate system parameters that are measurable. That's pH, dissolved inorganic carbon, or DIC, total alkalinity, TA, and PCO2, which I was talking about, the partial pressure of CO2. So the CO2 enters the ocean, and then I'm going through that equation with all the chemical species. And so that CO2 is then aqueous and reacts with water, and then becomes bicarbonate, and there's bicarbonate species. And what basically happens is that second equation, where when CO2 enters the ocean, it reacts with carbonate to form bicarbonate. And I'll explain why this kind of shift in CO2 species is important later. But for now, these 4 parameters are kind of what we can measure in the ocean. And with 2 of them, you can calculate the rest of the CO2 system. Okay. And I'm going to go into more detail in each of these parameters next. Okay.
So do you want the next slide, Sophie?
Yes, thanks. Okay, so pH is defined as the negative log of the hydrogen ion concentration. It is dimensionless, and in that figure you can see, so an issue with pH is that we say that, you know, seawater pH has changed from pre-industrial times from 8.2 to currently 8.1. And, you know, in your mind, 0.1 change in anything doesn't seem like a lot. But because of the log scale, it's actually a pretty big change. If you look at, you know, that left side of that figure where it talks about, you know, the tens of per unit of pH change, 10 times, you know, from one unit to the next. So you can see, you know, different things that we're familiar with, you know, for low pHs. So it's also, you know, that definition is negative. There's a negative correlation there. So lower pH is more acidic. You have a neutral pH at 7, and then anything above 7 is a basic. So things like Coke and lemon juice are acidic. You know, people say you drink too much soda, it rots your teeth. It's not just something to make you not drink soda, but it actually is damages the enamel in your teeth, because your teeth are made of calcium carbonate. which I will talk about later as well. And things like soap are basic. So yeah, we can move on. So in the ocean, surface pH is an average of 8.1 currently. It ranges from 7.7 to 8.4, so it's slightly basic or alkaline. pH is a pretty easy measurement to make, but difficult to make it very accurately. And there's several different conventions or scales that pH is measured on. And we can go into that more in detail later if anyone's interested. pH is lower at high latitudes and in upwelled waters because pH in deeper waters is lower, and then when it's upwelled, it brings up that lower pH or more acidic pH water. pH is directly affected by temperature because it is related to CO2 gas solubility, and that is directly affected by temperature. And there's lower buffer capacity in high latitude oceans versus low latitudes. Okay. Next slide, please. So the next thing is dissolved inorganic carbon, or DIC, or people also call this total dissolved carbon dioxide. There's a lot of names and they all mean this definition. So I talked about earlier bicarbonate, which is that first chemical species that's 90% of DIC. There's carbonate that's 9% and 1% is dissolved carbon dioxide. So total dissolved inorganic carbon is, the sum of all those species. And average DIC in seawater is about 2,000 micromoles per kilogram, to give you an idea of what that measurement is in the ocean. Also, if there's any questions along the way, please feel free to unmute yourself and ask, or put that in the chat. And then Alexis, if you can let me know if somebody, I'm not totally paying attention to the chat, but
yeah, so actually during the presentation, can you use the chat box? That way we'll have time at the end to do more interactive question and answer, because we want to save a good portion of the time for that. So please do type any questions you have in the chat box. And for those who recently joined, one question I'm going to pose to everyone is for you to tell us where you're interested in monitoring ocean acidification, and just tell us a little bit about the site. What is it near? What are the conditions like? Are you already monitoring there? And that will give us some really interesting things to talk about in our discussion. Sophie, do you want to move on to the next slide?
Yes, please. So DIC in the ocean ranges from 1800 to 2250 in the open surface ocean, and the average surface ocean value is 2075. This in contrast to pH, which is easy to measure but difficult to do well. This is difficult to measure, but if you can measure it, you can do it pretty well. It is highest in the high latitudes. So that when I say high latitudes, I mean like the northern part of the ocean, in this slide, the highest north and south, because latitude is horizontal. And then DIC can be affected by CO2 solubility as well, and that relates to temperature and biological processes. Okay, next slide. So total alkalinity is something that people have a lot of difficulty wrapping their minds around. And the way that is easiest to think about is proton acceptors minus the proton donors. So the amount of base you have minus the amount of acid you have. And this is I'm not going to go into buffer capacity, but we can talk about that later. In the ocean, carbonate alkalinity, so that's bicarbonate and carbonate. So because there's a 2 in front of that carbonate, so that when I say carbonate, I mean CO2 3 2 minus. That because there's a 2 minus there, so you're counting that twice because it's doubly effective in absorbing a hydrogen ion. So that carbonate and bicarbonate is 96% of the total alkalinity. So a lot of times if you're trying to do a quick estimate of alkalinity and you don't have measurements of these other ions, so chlorate, silicate, phosphate, then you can use that estimate of carbonate alkalinity to estimate how much total alkalinity there is. It can be easy to measure, but also difficult to use if in certain regions where there are higher contributions of these other ions besides carbonate and bicarbonate, especially in coastal regions where many of you might be doing monitoring of OA. The species that are below that red box in that table can be more important. Okay, next slide. So alkalinity in surface seawater varies between 2000 and 2450, and average alkalinity is 2300. It's also can be a difficult measurement to make and difficult to do well. So, and alkalinity is often estimated from a relationship with salinity. This is something that you can do often in the open ocean. Different coastal regions have different inputs to salinity, so sometimes you can't predict alkalinity as well in the coastal regions from salinity. It is mostly conservative in the open ocean. And okay, yeah.
I'm just going to add a note here, Sophie, which is that I think, you know, when we talk about monitoring OA, we recognize that some of the standard techniques are quite expensive to use. And so something that Burke, Sophie, and I are very engaged in is trying to lower the cost. And one of those areas is actually trying to tell you, you know, if you have access to a lab in your country that maybe has really good analysis equipment to analyze the alkalinity, and you have the ability to look at salinity, you can establish what that relationship is locally. You just need to have enough data. But you can think about, like, there are sort of these cheats you can do, but you're going to need to do a little work ahead of time for them to be valid. So that's just an example, but I think we can give you other ideas of ways to kind of lessen the cost or the complexity of some of these measurements by perhaps partnering with another lab that can help with some of the analysis or changing the way that you're doing things. So that's just one example. So if in Nigeria, you know, there's one lab that's going to be doing the TA analysis, people from all over the country might be able to submit samples for a certain period of time, and then they might understand their salinity alkalinity relationship. So just noting that.
Yeah, if you want to make a alkalinity salinity relationship, you're gonna need to have, you know, samples for both of those things that cover seasonal changes. So if you live by a river or something like that, that will change the salinity, then you kind of need to cover when that river is going to be, you know, higher volume, putting fresh water into your region of study. But yeah, we can go into that. I'll be done with this part soon, and we can go into more monitoring aspects. Okay, next slide, please. So talked a little bit about this already, partial pressure of CO2. And that is, you know, so PCO2 is dissolved CO2 gas, and that what that pressure is. So, you know, gases are measured in how much pressure they exert. This is a scrap what I'm just saying, but it's the pressure of the dissolved gas in the water sample, and it has to do with CO2 gas solubility, which is temperature, and mostly temperature dependent and also salinity dependent. And there's this constant called KH, which is Henry's law constant. That is the gas solubility, and then the ratio of that with the concentration of CO2 in the water is what gives you your PCO2 in the seawater. So there's a lot of different units to measure partial pressure of CO2. It is what we're going to talk about is you might have heard ppm, which is parts per million. And that is technically the mole fraction of CO2, which if anybody is interested, we can go into later. There's small nuances basically in all these different ways of measuring or reporting CO2. Okay. Move on. So PCO2 in the open surface ocean has a pretty large range. It varies between 150 and 550 microatmospheres. And the average surface value is about 400. And that is basically, you know, the surface ocean is in equilibration with the atmosphere. And so the atmosphere is about 400 or a little slightly over 400 microatmospheres. And so that's reflected in the surface ocean value. There's a lot of different sensors that measure PCO2. And the question is though, how well do they do it? And how expensive is that equipment? So for you guys, PCO2, well, Burke is going to talk about what parameters are best to measure later. So I'm going to not talk about that. The PCO2 is strongly affected by temperature and biological processes. Okay, next slide. So those four parameters, PCO2, alkalinity, DIC, pH, those are what you can measure. And then this is something that can be calculated from those measurements. So carbonate saturation state is a measure of, there's a lot of organisms in the ocean that make their shells or skeletons out of calcium carbonate. It's a mineral that is calculated by the concentration of calcium and the concentration of carbonate in the ocean. And it basically tells you how easy it is to calcify in that water. And there's, so if the saturation state is equal to 1, that means it's in equilibrium. So, and then below 1, it's very difficult to calcify and you might be experiencing dissolution. And then over 1 is best for, you know, if so, corals are very sensitive to aragonite or carbonate saturation state, and they require saturation states of greater than 1 to build their skeletons. And so there's, I was just saying aragonite because there's 3 different forms of carbonate, calcite, aragonite, and magnesite. And calcite is the least soluble and magnesite is the most. Oftentimes in the open ocean, there's these organisms called pteropods, and a lot of them form their shells from aragonite, and that's kind of an indicator species for ocean acidification. And it's more sensitive than calcite, so a lot of studies use aragonite. Oh yeah, I also, the concentration of carbonate is what kind of drives this saturation state, because the concentration of calcium in the ocean is pretty equally distributed everywhere. So it depends on the carbonate concentration, mostly. Okay, next slide. So in the open ocean, the calcite saturation state varies between 1 and 6, and aragonite is more soluble, so that's about 40% lower. Average for calcite is 4.5, and for aragonite is 2.7. Like I was saying earlier, this can't be directly measured because it depends on concentration of calcium and carbonate. It is very sensitive to the ratio of alkalinity to DIC, which can kind of be thought of as the buffer capacity. And it's not as sensitive to temperature and more sensitive to salinity due to its dependence on the calcium concentration. Okay. Now,
one thing I was going to note here, for those who might have attended Sam's lecture yesterday, you probably talked about the biological significance of the saturation state. We talk about 1 because, you know, at a saturation state below 1, basically the calcium carbonate is going to dissolve. But for many organisms, the threshold is a lot higher than that. And particularly in coral ecosystems, you find that coral really prefers a saturation state around 3 or 4. And part of this is that where the organism is doing their calcification, usually they have some sort of organelle or internal calcification process where the organism is elevating the saturation state in that organelle to make it easier and easier for them to build calcium carbonate. And so, you know, it becomes more energetically expensive for that process as the background saturation state lowers. So don't just think about 1 as the number to be aware of. Different ecosystems have different thresholds, and different species have different thresholds. So one common area of study is sort of correlating the saturation state with perhaps the shell composition of organisms. So there's been some really interesting research cruises in the US, like off the west coast of the US, where they've kind of collected all of this chemistry data and also looked at pteropod shells or crab larvae and done some comparison. So just to flag, you know, this is one of the parameters that is of most interest for biology. Of course, you can't measure it directly. Like Sophie said, you have to measure two of the other parameters to get this. But just to add a little more context.
Yeah, two more points. One, if you look in the figure, you can see the redder colors are higher saturation state, and that's where most of coral reefs in the ocean are. So, you know, corals definitely like higher saturation state. And second, a lot of research, there's been a lot of research on the West Coast on ocean acidification and its effect on biological organisms. And so on the West Coast, there's summertime upwelling, which brings that low pH, low aragonite saturation state water up to the surface. And there are lots of studies on when that exposure during the life cycle of a Dungeness crab larvae or a pteropod larvae is going to be, so, sorry. There are studies on if, you know, pteropod is exposed to aragonite saturation state below 1 during its growth stages. You know what I'm trying to say, Alexis? Like,
Yeah, so the life cycle, the stage in the life cycle at which the exposure happens is often a determinant
important
factor.
So if it's exposed when it's just a larvae, then it will probably die because it doesn't have any buildup of energy stores to, you know, use up. But if it's exposed when it's already an adult, then it might be able to survive, but its reproductive capacity might not work, you know, might be lowered.
So another kind of angle to this is that, let's say you have a species of high importance in your ecosystem or your, maybe for economy, if there's a particular shellfish or shell building organism that's very important, you might want to study how it's at different stages of its life cycle, it's affected by something like saturation state, and it can actually inform management decisions. So what if you have a particular shellfish species that's very important as a food source and you find that it's very vulnerable in its larval phases? That actually makes the case for having a hatchery for that species, right? Because in a hatchery you're able to control the chemistry during those sensitive early life stages. And so this is where, you know, this science can really plug into management, and you can talk to fisheries management, you know, experts and others if you have some information about, well, you know, in this bay we're seeing this sort of trend and we anticipate that these are the conditions that these organisms are going to experience during the juvenile stage. So therefore, we strongly recommend that the government consider investing in a hatchery for this species, or something like that. That's just an example of how you can really apply your research to management.
Okay, now I'm gonna hand it off to Burke.
All right, well, thanks, Sophie, for that great setup. The thing that I want to try to emphasize here is that carbonate system is inherently a multi-stressor system. One perturbation to the system makes many aspects of carbonate chemistry change, and there are different physiological and life stage responses to each of those parameters. And there is a lot of conventional, quote unquote, knowledge about how the system behaves. And so there are a number of things that we want to make sure that are understood, and some sort of dogma that we want to dispel. So we've often heard terms like pH is a master variable, or that the PCO2 of the water is in equilibrium with the atmosphere, or that The mineral solubility will determine the carbonate chemistry, and many of these things are wrong in the real ocean. It turns out that pH really doesn't control CO2 chemistry. CO2 chemistry controls pH. In terms of things like gas exchange or mineral precipitation and dissolution, the system can often be far from equilibrium. And the other thing to remember is that the equilibrium between the acid-base species, so that first set of equations that Sophie showed, is so rapid that the instant you change one part of the carbonate system, for example, by raising the PCO2 in the atmosphere, all of that equilibrium reactivity happens and redistributes the perturbation in ways that can be hard to predict. Only total CO2 and total or titration alkalinity can actually quantitatively track additions or subtractions from the carbonate system. And these are really what drives how the system responds. Next slide. And what's important to think about is how the responding species react in the face of changes in total CO2 and alkalinity. pH goes down when total CO2 goes up. It goes up when total CO2 goes down. Alkalinity has the opposite effect. pH has got strong temperature effects and also complicated salinity effects. PCO2 goes up when total CO2 goes up, goes down when total CO2 goes down, and the opposite in response to alkalinity. It's got a fairly strong temperature effect, but it's very well understood. And the salinity effect is dependent in some respects on what fresh water is driving the salinity change, but it's very well known. Aqueous CO2, which is directly related to PCO2, has got very similar behavior to PCO2 itself, but it's got a much reduced behavior with respect to temperature because it does not depend on the gas solubility. Omega, it's got strong dependencies on TCO2 and total alkalinity. It's fairly weakly sensitive to temperature. It's sensitive to salinity, but again, in ways that are easy to understand. The thing that is important is that you can't assume that a single perturbation is going to affect all of these species in the same way from one location to another. And this is especially true in coastal settings. The lower plots show omega as in plotted next relative to pH and PCO2 from a coastal setting off the Washington coast. And what you can see is that even for a given pH of say, 7.8 or 8, there's maybe a factor of 2 or 3 range in the omega that corresponds with that pH. Next slide.
Sorry, I'm responding to a chat. There you go.
The other thing that is important to remember is that now that we've talked about how the individual parameters respond to changes in the bulk parameters, total CO2 and alkalinity, then it's important to think about how those bulk parameters respond to environmental forcings. Total CO2 and alkalinity are conservative with respect to temperature. They do not change as the temperature changes. They do not change as the pressure of the sample changes. So the measurement that you make of total CO2 and alkalinity in your laboratory at your laboratory temperature is exactly the same as those concentrations existed in situ, even if the water was much colder or came from a great depth. Salinity impacts these two species, but in, again, straightforward ways that we can constrain. Photosynthesis drives total CO2 down strongly, drives alkalinity up weakly. Respiration drives total CO2 up strongly and alkalinity down weakly. Gas exchange impacts total CO2 significantly, but it has no effect on alkalinity. That's an important thing to remember when you're talking to folks outside of the scientific community when they suggest that the ocean is becoming less alkaline as a result of rising CO2. The alkalinity is not changing. It's the distribution of the acidic and basic species within seawater that are changing in response to the PCO2. Alkalinity is not affected by gas exchange. Calcium carbonate processes impact total CO2 strongly. Precipitation drives total CO2 down. Dissolution drives total CO2 up. Alkalinity has got an even stronger response to calcium carbonate precipitation and dissolution because for every one of those carbonate ions that is taken up or released, there's a doubling effect on alkalinity. Next slide. So what does this mean in terms of measurement and monitoring? And this is unfortunately equivocal. I would love to say that there's one best way to do things, but that's not really the case. And it depends not only on your capabilities and your budget, but it also depends on the system you're working in. The open ocean in blue water is a very well-behaved system. Alkalinity is fairly easily to define. Dynamic ranges are not large. Salinity ranges are small. And this is not the case in almost any coastal setting, and certainly not where you're interacting with dynamic systems like river plumes, mangroves, coral reefs. So what you need to ask yourself is, what do you really want to know? Do you care most about pH? Do you care most about omega? Do you care most about dissolved CO2? And then you need to ask yourself how well you need to know it. Do you need to know omega to plus or minus 0.01 or to plus or minus 1? And either of those answers can be completely valid depending on the system you're working in. How fast do you need to know it? How much do you have to spend? And how much technical skill do you or your facility possess with this regard? So a few things are certain. Regardless of the answers to the questions to the left, you always need to measure temperature and salinity. And you always need to fully constrain the whole carbonate system. The reason for this is that a single measurement does not tell you about the other components in the carbonate system with sufficient accuracy or certainty. And you don't often know which parameter in the carbonate system your subject species are responding to. There are a number of instances where we know pH has an effect on metabolism. But there are a number of instances where we know pH doesn't appear to have an effect on shell formation, and it's omega that matters first. The best advice is to try to combine one of the two, what I would call conservative parameters, either titration alkalinity or total CO2, with one of the more sensitive parameters, which would be pH or PCO2. It's so critically important to have a full understanding of the characteristics of your system and the potential complications with your measurements before you dive into this, because if you're working in a system that has got a lot of what we would call non-carbonate alkalinity might be a bad measurement for you to make. If you're working in a system that has a lot of temperature and salinity dependence, pH might have uncertainties that could be confounding. Next slide. So you also really want to think about what processes you're interested in, right? And so if you're interested in gas exchange or calcification or metabolism or mixing or anthropogenic carbon, total CO2 is essential. If you're not so interested in gas exchange or anthropogenic carbon, but you really want to learn about calcification, alkalinity is a great measurement to make. pH really depends on what you're after, and there are in many instances at current values of ocean pH where we are so far from direct metabolic effects on organisms as a result of lowered pH that it may not be that relevant in context of the processes or the effects you're seeing. PCO2 is obviously the driver of gas exchange. It's a sensitive indicator of calcification. It has a potentially large effect on metabolic and motility functions physiologically, and so you might want to do that. Omega is absolutely critical if calcification is the process that you're interested in studying. Next slide. And I won't go through this table in detail, but in order for you to constrain the carbonate system, you need to make 2 measurements of the carbonate system chemistry in addition to your temperature and salinity. There are 4 measurable parameters, which means there are 6 combinations. And depending on what you want to know, some parameters are extremely useful. If you want to know total CO2, measuring total CO2 is the best way to do it. If you want to know PCO2, measuring PCO2 is the best way to do it. If you want to determine pH, it's not necessarily true that measuring it directly is the best way, because the precision of the measurements can be so challenging to achieve, and the uncertainties related to scale and salinity dependencies can be very confounding. And so, for example, if you want to know pH, it might be more useful for you to measure total CO2 and PCO2, because the calculated pH from that parameter pair is very good. It's very robust in the face of changing state variables like temperature and salinity. And so just think a little bit about your capabilities, which parameters you really want to know, and consult this table and say, well, you know, what I really want to know is pH. And so therefore, my measurement pair should not be total CO2 and titration alkalinity, because the errors associated with calculation from that compound rapidly, and the uncertainties in your pH can be very large. Next slide. So that's what we have, and I'm happy to take questions. We should probably let Sophie field questions first, and interested to hear how your situations and your systems relate to this.
Thanks so much, Burke and Sophie. And I also wanted to let everybody know, I put a question in the chat box, and if you can't see the chat box, I'm going to read it for you. So essentially, we would love to learn more about where you are hoping to do ocean acidification monitoring, or maybe where you're already doing it, because that helps us give you some advice. And I think we can each learn from each other's sites. So, you know, where are you interested in doing this? Is this near a mangrove ecosystem? Is this near a river? Is this really near shore? Is this offshore? How variable do you think this system is seasonally, or even within, you know, the tidal cycle? We would love if you type your description of your site into the chat box, and I think that'll be a great way for us to just kind of give examples of how you would go about monitoring that system. And so we do have a couple questions and examples here that I'm going to read out. Please, if you have a question, type it in the chat box, or you can also raise your hand using Zoom. And then we will do our best to answer those questions. And I will let everybody know that, number one, this is being recorded, so you will have an opportunity to kind of re-listen to things. And number two, if you want to have a really kind of in-depth conversation about your site, please apply for a coaching session. That's the purpose of these coaching sessions, is to have just a long conversation only with you and your lab and the trainers. And so if you have any questions about how to apply for the coaching session, email me. My email is associated with the announcement. So with that, I'm actually going to read a few of the really great questions and answers we already have in the chat. One that Stephen posed to everyone is, and it's a very good point. So for most organisms, it's really the variability, the natural variability that the organism is exposed to is extremely important to know, both in terms of understanding how sensitive is this organism to change, but also to design a good experiment, you need to know what the current variability is, or else you might not be studying something very interesting. So for example, if your ecosystem is already experiencing a swing in omega of, you know, 1.5 to 3, that would be pretty extreme, but if it's experiencing that, you know, because of just natural processes, studying in a lab the difference between 3 and 1.5 on that organism almost means nothing, because that's part of their natural variability. It doesn't really tell you anything about the future. But first you need to know what the natural variability is. So what Stephen was asking is, how well is the standard deviation of OA parameters known at the relevant timescales across the world oceans? So I took a stab and said it was pretty well understood in the open ocean and much less well understood in the coastal systems, which is why local monitoring is so important. But if Burke or Sophie want to add their thoughts to that question, I thought it was a great question.
So the parsing of that question is, what do you mean by the whole ocean, and what do you mean by relevant timescales? So if we talk about the whole ocean as the global ocean, and if we limit our discussion to the surface global ocean, then we know quite well what the variance in time and space of many of those parameters are. Extensive extensive measurement programs, detailed analysis. Certainly get the Takahashi marine chemistry paper from 2015, from which several of the figures Sophie presented or showed. And that really does a fabulous job of comprehensively showing what's happening there, both in terms of season and location. The problem is that the reason that you can't answer this so straightforwardly is that the timescale is one, and for example, our work with larval shellfish is an example, and their sensitivity to omega is intense within the first few hours of their life, and there are very, very few places anywhere where you've got hourly resolution measurements of carbonate chemistry. Further, the variance in coastal systems tends to be much larger and not as simply extrapolatable from one location to another. The behavior of carbonate chemistry in Yetarts Bay off the Oregon coast or on the Oregon coast is completely different than the behavior of carbonate chemistry in the Mississippi River plume, for example. And so we really need to know more specifically about what is the timescale that matters to you and your problem, your organism of interest, and what is it about your local system that you can explain that helps us narrow that down. Sophie, do you want to add to that?
Yeah, I think all those points that Burke and Alexis made are really important. Something about coastal systems is residence time. So how much that water is sitting in that bay, how long that water is sitting in that bay is really important to consider. I did some work in my PhD studying salt marshes, and we did see, almost in certain times of year, the pH would change by a whole unit over one tidal cycle. So there, Alexis, you were saying what a saturation state change of 1.5 to 3, like that is possible in certain places. So, and the point that Burke made about one coastal system varying very much from another is really, can't emphasize that enough.
Yeah, I want to give the Sophie and Burke an example to respond to. So we actually have a couple of people who are interested in studying this around mangrove ecosystems. So we have, let's see, I'm scrolling through the chat. So in Nigeria, there's a monitoring team at the Nigeria Maritime University who are interested in the Niger Delta. And this is a mangrove-dominated area with a lot of creeks, estuaries, and ocean. And it's also where oil exploration and exploitation occurs with a lot of gas flares. Then we had someone else saying, what is the expert opinion about the mangrove ecosystem monitoring and OA studies? And so maybe Burke and Sophie, just as like a scenario, right? Let's say you're starting a monitoring program in an area with lots of mangroves, also high variability in terms of freshwater influence, and you have these external things happening. Where would you begin? How would you start? What would you do? What methods would you use? Just like walk us through it.
I also want to say that even within like one system, there's going to be a lot of variability in like different pockets in that one system. So where you're going to try to have your monitoring station is going to be really important to think about. I think, yeah, I mean, can we speak like ideally, if you have a lot of like unlimited amount of resources, like what?
No, I think we should think about limited resources. If you have limited resources, how do you begin, right? This is a question we get all the time. Where's the first site where you should be monitoring? and how do you even begin to prioritize where to start monitoring? So if you have a really complex system, you know, where do you begin?
So the only thing I can say in a really complex system is that you have to focus on what matters most to you first. And so Sophie's absolutely right. Within an estuary, within a bay, within a coral reef, you can find extreme departures from sort of mean behavior in time and space. But if you're working on a system where you're trying to establish an aquaculture facility, then sample your intake, right? Sample where your shellfish rafts are being placed. That's the only thing you can do without comprehensively and exhaustively sampling. Some of these places, mangroves, for example, that was a question that was put out there initially. Mangroves can be really challenging for pH and alkalinity measurements because of the contribution of organics and our sort of lack of understanding of that. And so even though total alkalinity or titration alkalinity and pH may be the more accessible measurements to make early on, they can be very, very hard to interpret unless you make a lot of other measurements, including the dissolved organic species and maybe even their acid-base characteristics. And these are areas of cutting-edge research, not service level measurements. These are very difficult things to do. And so even if your system is one of these, and even if you don't have the capability to make the measurements right away, starting a sample collection program that helps you to establish some sort of a baseline. And again, I can't tell you without knowing more about your system what the sampling resolution needs to be. If you have a system that's strongly tidal, you need to sample at sort of twice the tidal frequency. If you have a sample that's a system that's strongly seasonal, you need to probably sample every couple of weeks or so. And so I don't know how to define that as an answer for everyone. And you know, it may be one of these things where we set up a, you know, an AMA or a FAQ sort of a site, because it's hard to know exactly what type and what distribution of circumstances we're in.
Burke brings up a good point about being able to ask questions later. So I want to make sure everyone's aware of this, a couple of resources. I'm going to type them into the chat. So the Global Ocean Acidification Observing Network is a network that's been established to kind of organize and set best practices for this type of work. And they also have a mentorship program called the peer-to-peer program, where you can sign up to receive mentorship from someone with more experience in this field. And you can actually apply for scholarships through the Ocean Foundation to receive some funding to maybe purchase some equipment, or maybe have your mentor come to your lab and help you set something up. Another thing is something called the Ocean Acidification Information Exchange, which is basically a forum that is specifically for questions about ocean acidification. And when you have a question, you can pose it in the forum, and different people can provide feedback on your question. And then finally, like I said, the coaching sessions are going to be an opportunity to get a lot more information. Let's see, I'm going to try and read through the chat and see what else we have as far as questions. And then if you want to ask a question with audio, if you can let me know in the chat, or also use the raise hand function, and we can unmute you and get your question in.
I have two things to say. One, like Burke said, some of these things are, you know, like cutting edge research that, you know, people with lots of resources can't even really answer that well. So, you know, that's, you know, the technology that we have currently still can't answer some of these questions. So I think just something to keep in mind. Secondly, that question from Margaret about natural variability. How do you know if variability is natural or you know, anthropogenic or something like that? There are these, there's this phrase called time of emergence, And that basically it's telling you, it's kind of a more recent concept, and basically it tells you how long you need to observe at a location before you can tell what that natural variability is. And for some places, it's like 30 years that you have to have observations to be able to tell what the difference is between the natural variability and the anthropogenic portion. So there's this thing in GOAN, the Global Ocean Acidification Observing Network set. There's these different levels of data quality. There's weather quality, which can tell you short-term variation. And then there's climate quality, which is telling you these like long-term trends in variability. So the long-term, the climate quality is something that has to be super accurate. And that is already, you know, there's very few sensors that are commercially available that will even measure that quality of data. So that is a pretty, you know, difficult question for everyone, you know, thinking about how to parse apart the natural and anthropogenic signals.
This brings up another question we got in the chat, which is about how do you monitor, like, for example, pH? And maybe you can talk about the difference between using sensors and discrete samples, and both like the costs, the ease, and then like what you can say from that type of information.
So we've talked about this a fair amount. I was involved with the X Prize activity, trying to push new and more affordable pH measurement technologies forward. Again, we need to know how well you need to know things. We joked during the X Prize competition that if I had taken a pencil and written 7.8 on the pencil and called it a pH sensor, I would have won the competition, because all of the samples were about 7.8, and the competition was analyzed based on stability and repeatability and speed of response. But you're working in systems that are very different than the open ocean. You probably don't care about a pH change at 0.001. And the question is, for your problem, for your area, what new information is valuable? And I think it's a failing of the research community, the scientific research community, to suggest that if you can't measure pH to 0.001, that it's not good enough. If you're working in a system that you know is dynamic, because you can look at the water and see it change from brown to green, from turbid to clear, you know that the tides are large, you know that there's a strong monsoonal effect, and you know that there's no data. The value of new information, even if it's of lower quality, as long as you have an idea of what that quality is huge. If you've never seen any data on pH or omega in your system, and it doesn't exist, and your system is dynamic, knowing that whether your system sits at pH 7.8 versus pH 7 is important new information. And so you shouldn't be discouraged by some of our tendencies to insist on demanding rigor in your measurements for areas that are dynamic and understudied. So how would you make those measurements? Again, in these circumstances, the most robust measurement pair is total CO2 and PCO2. And often labs don't have that capability, but there are labs globally, regionally, that can make those measurements. And so the first thing to do might be to just start a sample collection program and send samples for analysis so that you have an idea of what the circumstances are like. pH electrodes off the shelf are risky. They are processed, the signal is processed in ways that the manufacturers don't fully divulge. There are just an awful lot of ways to look at that. However, if your pH measurement is uncertain to plus or minus 0.2, which is massive in the context of the global ocean acidification signal, but you've never made a measurement of pH in your system, plus or minus 0.2 might not be too bad. It's a great way to start, and you have to start somewhere.
I learned that we have a guest who's on their phone. Andrew Dixon is here and on his phone. So if there's ever anything, Burke or Sophie, that you want Andrew to answer, I have to manually unmute him. So let me know.
Don't ever do it.
So just let me know if we should unmute him. I think to go on Burke's thing, and I think some of you know about this, but we've been involved in a project that Burke and Sophie have helped with a lot. Which is the Go On in a Box system, which the Ocean Foundation sort of started developing because we recognize that so many places they need to know where to start, and we need to give them a good system that will get good enough data for coastal observing. And so this box of equipment, it costs about $20,000 US for everything, which sounds like a lot, but it's a fraction of what the systems being used, like in Sophie's lab, are. And it will get you what we call weather quality data. And in fact, we're actually seeing climate quality data when Chris's students, Chris Sabine, who spoke on Wednesday, he had a high school student run samples on this and get climate quality data. So we feel pretty confident in this system. The list of equipment that we recommend is publicly available on the Go On website. You can also ask me. And then whenever I get funding in for this, I try to support the purchase and distribution of these systems to labs around the world. And so there are recommendations for how to do this well at a lower cost. So don't feel like you have no resources, right? We've thought a lot about this. We've got some good recommendations to get you started. And a lot of what we recommend is sample collection and analyzing those samples in your lab, because that's something you can do relatively cheaply that's going to get you pretty high quality results. Now, knowing how often to sample and where to sample, those are in a ways the most difficult questions to answer. But like Burke said, you have to start somewhere. I got a text from Andrew and he said, thanks, Burke. So we have a couple other questions. So I know Harriet a while ago asked, or she described her site, and she said they're monitoring open ocean along the eastern and central coast of Ghana, and this stretch of coastline has a number of lagoons and rivers which discharge into the ocean, and they're highly productive for fisheries. So, you know, in that situation, I think, again, the question is, what do you want to know? What is most important to you? You know, in some cases, we recommend people, if there's absolutely no data at all, to start in a place with the least variability, to just get some sort of baseline of kind of what is quote unquote normal. So you might want to start your monitoring away from these rivers, because that's just a more complicated place to study it. But if you're mostly interested in something that grows right along those rivers, then you might want to study there. I don't know if Burke and Sophie want to add anything to that. Okay, yeah, I passed the test. And then we have another question from Falulu, who, by the way, is a recipient of a peer-to-peer scholarship. So really important resource for you all to know about to maybe get some funding support. He's studying coastal OA in Western Nigeria, and field studies are quite challenging. However, laboratory biological studies can be well constrained. And in line with this, they will be working together at the Nigerian Institute for Oceanographic and Marine Research, and there'll be a team doing biological laboratory studies. And he's going to apply for the one-on-one coaching. So I think that's a good example of a lab that's trying to start somewhere, you know, and get some interesting information. I will caution that it is important to know your natural variability to do a well-designed lab experiment. And there's ways that you can I mean, I don't, Burke and Sophie can cut me off if I'm going way off the deep end, but I think that if you're studying something like biologically, even understanding the variability and the salinity of where that thing lives gives you information that can inform the design of your study. It's not going to be the most sophisticated information. It's not going to like exactly tell you what the carbonate system variability is, but it might give you an indication of variability, which will maybe tells you if you need to study it better before you can design a good experiment. So Burke and Sophie, feel free to weigh in if you think I'm totally off base there.
No, I think it's good. And what Burke said about always measuring temperature and salinity is really good. It can give you a lot of information just from those two parameters. And then if you can get, simultaneous bottle samples, at some frequency, and see how temperature and salinity change with some carbonate parameter, hopefully 2 carbonate parameters, then you can kind of have an idea of what's going on. I was going to say something that, so coastal systems have really high variability just from one tidal cycle to another, and so you don't, if you have a electrode that has a uncertainty of 0.2 units, pH units, then you can still see that high variability. You can still capture that high variability, even if your electrode is not a super accurate.
Yeah, some data is better than no data. And with biological studies, knowing that natural variability is very important to designing a very good study. So if you're interested in the biological studies, and Sam has a ton of resources on this, and his recommendations for exactly how you do this. And I think Sophie's right. Sophie and Burke are right. The pH electrode, you're not going to want to use that to tell anybody what the carbonate system is like in your area, but it can inform your decisions about how frequently you're going to monitor, or with higher quality systems, or it can just help you capture the variability. So there's going to be the recommendations that you see maybe at Sophie's lab in Seattle, what she's doing, which is like assuming kind of like the best resources, but there is so much else you can do. You just have to understand the limitations of what you're doing and what you can and cannot say with that data. And if you don't know where to start, that's where there's a really wonderful, I think if there's nothing else you take away from this week, it's that we are very friendly in the OA community, and we're very interested in helping people start their work. And so we're very collaborative, and we always welcome questions. And we're very happy to help you answer some of these questions, because it is so dependent on your individual ecosystem that you want to study or the species that you care about.
And super hard to, come into this field and not know where to start and feel like super overwhelmed, but just, one step at a time.
So please, if anyone has any questions, again, I can unmute you if you have something you want to ask the trainers yourself. So just let me know in the chat, or you can use one of the buttons in Zoom to kind of flag yourself, and I'll unmute you. Otherwise, please type your question into the chat. In this downtime, Andrew, I'm going to unmute you in case you have any commentary. If you're not prepared, then you don't have to say anything. So Andrew, you are unmuted if you would like to talk.
Thank you very much, Alexis. I guess the only comment I'd make, which hasn't been emphasized so far, but certainly been alluded to, is that in the GOAN documents, this explicit difference between climate and weather. Realistically, the weather is looking to learn about ocean acidification at the kind of levels that may influence organisms. The climate is looking to see long-term changes as a result of anthropogenic activity. So realistically, I think most ocean acidification is at the weather level, but the difficulty is that most early work on the CO2 measurement community was at the climate level. And so there's real confusion about whether measurements with a larger uncertainty are necessarily useful. And as Burke said, yes, they're clearly useful. So don't even worry about whether they're useful or not. The only thing to worry about when you make the measurement is how uncertain is it, and therefore, how does that affect the kinds of scientific conclusions I can draw from it?
Yeah, very well said. Thanks, Andrew. I can unmute you, or I can mute you again if you'd like, or you can probably mute yourself on your phone.
Not easily. I
Oh,
tried
okay.
to unmute myself earlier, which is before I conveyed to you that it wasn't so straightforward.
Okay.
So it's up to you. If you suddenly hear a background noise, by all means mute me. At the moment, I'm walking in a very quiet area of my hometown, but that's not going to stay the same for the whole half hour, I suspect.
Thanks, Andrew. All right. Any other questions? Please don't be shy. Really, any question is fair game.
Okay, hello everybody.
Hi.
Okay, this is Abraham Percy speaking. Can you hear me?
Yeah, if you hold your microphone close, because it's just a little quiet, so hold your microphone close.
Okay, can you hear me?
Yes.
All right, thank you very much. Thank you for Sophie and Bob for the presentation so far. And Alexis, thank you very much for the presentation and section so far. I think some of the issues have been raised by my colleague Amara from also the Department of Marine Environment. Okay, I'm Percy Abraham. I'm a lecturer at the Department of Marine Environment and Pollution Control, Nigeria Maritime University. Okay, we are new to this. That's the truth. We've not started a monitoring program. We've not started anything about ocean acidification research or science. So we're just trying to see how to, learn all we can and start a program as soon as possible if we have the resources to do that. Okay. Open waters.
You just broke up a little bit. Can you repeat what you just said?
So can you hear me?
Yes.
Okay, I'm sorry. I think my earpiece is having issues, so I decided to remove it. Okay. I think we are surrounded by mangrove swamp, and it's as you move from there, you see the Escravos River, which empties into the Atlantic Ocean. I don't know. I have some pictures here. If you can permit, I would have said, okay, let me share. If you can share screen with me, let me just show some of the pictures. I put them in a PowerPoint presentation. Would that be okay? Some of the pictures of mangrove swamp. Okay, let me see if I can share screen. Okay. Okay, can you see what I'm sharing?
Yes.
Okay, good. This is like, because what we normally do is that we go on every week and come back home weekend. Most of us stay at the mainland and we'll go to the island to do our job. And, when it's weekend, we leave. So these are some of the pictures. As you can see here, this is the open waters of the Escravos River. And at the very front, if you go down, it empties into the Atlantic Ocean. And these are some of the other pictures. This is one of the islands. There's a campus of the university because we have two campuses. There's a campus on this island as well. And these, I'm just trying to show you the area as the open waters, and these are the mangrove areas. And here you can see this oil spill, film of oil spill on the surface of the water, very close to the mangrove swamp. Okay, these I normally take some of these pictures when we do a boat ride, a regular boat ride to the university. These are some of the staffs of the university going to work sometimes. packed full of both. So why am I showing this picture? Because we really want to start, just like you said. I think some of our friends from the Nigeria Institute for Oceanography and Marine Research, they want to start with maybe microcosm studies and stuff like that. But like you said, if we don't really have a good understanding of what's happening in the field, it will be difficult to design research in the lab. And for us, you know, this is where we work, and we don't know what is happening. So how can we start a monitoring system? As you can see in the mangrove, there's a lot of industrial activities. Oil spill is taking place. There's transportation. There's boat ride to different islands, and of course, the major island is Escravos, which is a crude oil terminal where Nigerian crude oil goes into the high sea and, of course, sell to the various parts of the world. So these are the peculiarity of our area. So, like, I want to know from what I've been said so far, where can we start monitoring? What is needed to start the monitoring, and how do we go about it? Because as I know, they've not been data and they've not been lab. I'm not certain there's any lab around that do major stuff like that. So from our background, I think some of us did some water quality research in the rivers and creeks and all that. And what we do sometimes, we collect samples, we put formalin or alcohol and take it to the lab for analysis. Sometimes we go with a HANA pH meter or salinity meter and stuff like that. So the question is that, can we translate that, or can we move from that stuff we normally do in the river into this saltwater system, and perhaps how do we go about it? So I don't know if you guys can really help us throw more light on this. Thank you very much.
Yeah, so what I'll say is that just any new information you can gather is really critical. And so even if it's, as Sophie said, temperature and salinity, that will tell us about the timescales of variability, the magnitude of variability. The processes that control temperature and salinity variability very likely impact the ocean acidification parameters, impact the carbonate system. If your background measurements include nutrients, nitrate, phosphate, things like that, those are process linked to carbonate chemistry and can be very, very useful in determining dynamic range, expected drivers. So any of that data that you can put together in a synthesis to say, we have a tidal signal, or we have no tidal signal, or we have you know, a river discharge signal that's related to seasonal monsoon, non-monsoon kinds of conditions. That's just really critical. Even if you have no carbonate chemistry data at all, it would be critical for defining what sort of carbonate chemistry parameters you want to measure, you want to pursue. The second thing is that it's very important to think about what you're trying to study. And as Andrew said, and as Sophie alluded, for you to determine how much ocean acidification has impacted your system in terms of a strictly anthropogenic effect is probably almost impossible. And so what do you need to know? Well, you probably need to know the mean and the you know, 10th and 90th percentile of variability in these systems. And you need to think about what system you anticipate being affected negatively or not. If you're talking about whether or not the mangroves productivity is changing as a result of anthropogenic impacts, then you need to go study the mangroves, and you need to choose a mangrove system that is impacted and a mangrove system that isn't impacted. And I'll come back again to saying that, you know, those are direct carbon system relevant measurements, and it's probably better constrained by using a carbon-based measurement, TCO2 or PCO2, than it is by doing pH. And if that's something that you don't have, but you know, could potentially collect samples and ship to Andrew's lab or my lab or Sophie's lab, we could get you some of the initial information about what we think that system is like. So that's really what I would suggest. Figure out what you think the appropriate scales of variability are with the measurements that you have, and then target a system and a process that you're trying to study.
Okay, thank you very much.
I would
don't
add,
know if you want to add something, or
yeah, I'm gonna add something there, that there's like two dimensions, right? There's like time how things change with time, and then there's how things change with spatial. So you can think about, you can do one experiment that kind of where you take samples, along the river.
Okay.
And then you can do one where you just stay in the same spot in the river and, take samples for every hour or two, for 12 or 24 hours.
Okay.
And that you can answer some questions with that, just, those two, like 2 days of samples. So, how the concentration is changing as you move along the river, which, is also going to be confounded by the river moving. But that will tell you, if you are moving at the same rate as the water, you're traveling with that water parcel, and is it changing from other inputs along the river as you go along, or just staying in one spot and seeing how the concentration changes, as the river is moving along throughout the day.
Okay.
And, that will tell you your daily variability, and then you can do that, like once a season or something, and that can tell you a little bit of how that daily variation changes seasonally.
All right, thank you. Thank you very much.
We had a question actually about freshwater acidification, which, might be relevant to rivers. So I don't know if Sophie or Burke, you want to talk about what freshwater systems and kind of how does acidification fit into that?
Yeah, so I'm happy to talk there, and Sophie probably can follow up. Freshwater systems can acidify just like ocean systems. They frequently are already at much lower pH than the ocean, and so the suite of parameters and sensitivities that you might consider could be different. And the other thing that you need to do, and I'll say this in general, if you're working in a system where your freshwater is a dominant factor, you really need to know what we call the freshwater end member looks like chemically. There are rivers, the average ocean river chemistry is sort of defined by the Amazon because it's the biggest river out there. And it has about half the alkalinity and total CO2 of the ocean. But the Mississippi River, for example, has got 50% greater alkalinity and total CO2 than the open ocean. And my guess is that some of the groundwater driven and evaporatively impacted rivers of the African continent diverge from those conditions as well. And I don't know a lot about those conditions, but it's very important to actually measure what the freshwater is, because assuming that freshwater just dilutes carbonate chemistry is generally not the case. The second thing is that rivers tend to carry a lot of the non-carbonate alkalinity species, and that makes the application of an alkalinity measurement in the absence of a number of other measurements to go along with it very difficult. And so you need to think about measuring dissolved organic material. You need to think about measuring some of the nutrient species, silicates, phosphates, ammonia. Those are all things that will contribute to the alkalinity characteristic of your river system, and will complicate your interpretation if you assume that the alkalinity is a simple ocean-like alkalinity.
After my PhD, I moved to the US.
Those are some great questions, everyone. Does someone else have a question they want to ask? Margaret had a question about sampling. So for such areas as Dr. Abraham's environment, what type of sampling is recommended, for example, along a transect or sort of mapping? I think, Sophie, you were alluding to this a bit.
Yeah, so I was saying along the river, like downriver, but you can also do across the river at different points in the river. A lot of times in rivers, there's, the water's moving fastest in the center of the river. So there can be differences across the river.
I think there's just a little bit of background noise from Taufik. Taufik, I'm just going to mute you real quick, but if you have a question, let me know. And we actually have a hand. Well, I saw Dimitri's hand raised, but now it's not raised anymore. Is Dimitri still here? Yeah, Dimitri, did you have a question? Maybe not.
I'm gonna just say it depends on what you want to know, right? So like if you want to know how the river, you know, what is changing in the river across a day, then you measure in one spot. And if you want to know how the river changes across the river in, you know, say at the mouth of the river where it, you know, comes out into the ocean, if you want to know, that can be a good spot to measure what the river is putting out into the ocean. If you just stay at that mouth, then, and take, you know, samples over a few hours over the day, then that would be interesting to see how the river output changes over a day.
And of course, seasonality is going to be really important for the rivers, right? And thinking about also the local industries, and if there's like a seasonal effect of those industries, like is there a time when farming intensity is a lot higher and there's more likely to be output of nutrients or other things? But the truth is some data is better than no data, right? And there's a lot of ways, there's a lot of interesting things you can learn, whether like you stay in one spot at the source, or like, you know, there's some places where they do like a long transect, maybe like a radiating transect to see like at what point the influence of the river is no longer detectable. And it kind of just depends on how much time you have to put into it and sampling it and all of that, and how you're going to analyze your data. I realize we haven't talked that much about the practicality of like how to take samples and like, you know, what materials you need. And I'll just like very briefly touch on that, which is that, you know, there's certain types of materials that we recommend using for the sampling bottles depending on which parameters you're studying. And this goes back to what Burke was saying about like how reactive different things are, or, you know, like for example, if you're measuring pH, you want to take that measurement as soon as you open the sampling bottle, because the pH is going to start changing as the air in the room interacts with the seawater, right? So there's all sorts of things that you can consider as far as what type of sampling bottles you want to use. Burke does a lot of work actually with beer bottles, and it's pretty easy often to get those types of bottles and be able to cap them yourself. One thing that is important to note is that the standard procedure for sampling always requires mercury to poison the water, because we need to stop growth. And so be aware of what the disposal practices are for mercury in your region. If there's like a particular procedure you need to follow, we have recommended procedures that we can share, including how to build something that will kind of concentrate it and allow you to filter your wastewater and just have sort of like a resin that you need to dispose of. But yeah, those are some other things. Sophie, it seemed like you wanted to add something as well.
I just forgot to say that another dimension you can think about is how concentrations will change with depth. So, yeah. I mean, there's like, so, you know, just in surface, there's differences in you know, like lateral space, and then there's vertical differences. So there's just, I mean, there's a lot of things to think about, and you have to just decide which question you want to focus on, and what you can measure the best with what resources you have.
Yeah, so, you know, we have a few more minutes. So if anyone has any final questions, you know, you can unmute yourself and ask, or you can put it in the chat, and we will make the presentation available as well as the recording. Any other questions?
Oh, not your question. Hello, hi. Thank you, Sophie and Hales, for the nice presentation. Now I have gained some insight into ocean acidification. So you just don't go and measure anything. So we have the parameters, what you are interested, pH, total alkalinity, and then the PCO. So you really need to understand the system, and then the tidal activities, low tide, high tide, because these parameters changes and they influence the ocean chemistry, isn't it? Yeah, so we are yet to start ocean acidification monitoring for Ghana. So when I will get in touch with you for further explanation and questions. So please send your email, and then please send a copy of your presentation. I like it. Thank you, Alex, too, for all the explanation too. God bless you. Bye.
Thank you all for joining. I'm going to put my email address in the chat. I will be on vacation next week. I'm going to a national park here, so I won't respond to next week if you email me, but I'll be back in the office. And Burke put his email in there too. And again, the coaching sessions, strongly encourage any group that's really wanting to start a program to apply for a coaching session, because then we'll do like some one-on-one work. We'll get at least one of the trainers you've spoken to this week to sit with you. And then there's other people beyond, like Sophie, Burke, Chris, Andrew, Sam. They're superstars, but we have a lot of other superstars in the OA community who are also happy to help, and we can help connect you to them depending on what it is that you're interested in.
I want to say one thing about monitoring. Write down the time and location of where you're taking your samples when you take your samples, and like kind of, you know, just for note taking. And, you know, you always, like, oftentimes when I go out and take samples, I'm like, oh, I'll remember this later when I'm, you know, when I'm sitting in the office. But there's like a sort of thing that happens to your brain when you're like in the field, and you're like, oh, I'll remember this later. But just write down everything that you can about like the conditions when you're taking your sample, and that will help you later when you're trying to figure out, you know, why something was a certain way, or if it seems like, you know, your data point is kind of an outlier, but you also wrote down that there was a storm the day before, and so you can kind of just basically write down as much information as you can when you do take samples.
I realized I forgot a super important thing, which is that there's the UN Sustainable Development Goals, which I think many of you are probably familiar with. One of the goals is about the ocean, and one of the sub-goals is actually specifically about ocean acidification. So it's SDG 14.3. And pretty much every government in the world said that they were going to meet these sustainable development goals, which means that your government has said that they will collect ocean acidification data and report it to the UN. And so there's somebody within your government who's the focal point for this indicator. And a great thing to do is when you start collecting data, make sure that focal point knows, and you might be able to get some support from potentially get some support from your government, because this is something that through an international agreement, your government has said they're going to do. There's also a really helpful template for submitting your data for this indicator, which we recommend using for any of this data. All of this information is available on the Go On website, and we can also share it with you, but there's a really great metadata template and template for submitting your data. And there's actually a database hosted by IOC within the UN where you can submit this data to have it hosted. So if you have any questions about SDG 14.3, but the nice thing is that all of your governments have said they're going to do this. And so this is a way for you to say, look, my work directly helps you meet this international commitment. And in some countries, you know, that has gone a long way in terms of getting some funding support or just getting recognition for your work. Well, thank you all so much. If there's any final questions, we can answer them. But the presentation and everything, I'll make that available. So,
okay. I think I have a question. Can I go ahead?
yes.
Okay. Normally, when we collect water sample in the river, like I said before, we normally use alcohol or formaldehyde for preservation of the sample and sometimes put them in ice pack before we get to the lab. But my understanding for seawater collection is quite different. Use mercury chloride or mercury. So I don't know if they can elaborate on that. And also, I was made to understand that you don't necessarily need to put the samples in ice pack or in a cold storage before you take them to the lab. So for instance, we want to send a sample for a very long distance. So I don't know what are the standard procedure we have to follow, and if there are special bottles for collection, and if there are other chemicals that can be used other than mercury chloride. Thank you.
Yeah, so again, with the caveat that what we're mostly interested in is this weather quality level of data. There are a number of things that you can do that are not as expensive or involved as some of the climate quality procedures. Alexis mentioned the beer bottle approach. We've been using that and have found that it's quite robust. I do believe there's a small alkalinity artifact in the beer bottles associated with the chemistry of the glass, but I think it's very small in the context of what we're trying to understand here. And we have a protocol. We can write it up and share it.
Great.
Yeah, and one of the things that we like to say is that oceanographers are really, really good at making empty beer bottles. And so beer bottles are fairly easy to come by. And because of the rise in home brewing, it's very easy to find caps and capping tools. The other thing that we found is that using a plastic dropper bottle for your mercuric chloride solution gives a very precise, very repeatable 40 microliter addition. And so you don't have to bring, pipettes, which might be several $100s, into the field. You don't have to bring the solid mercury, which reduces the contamination potential. And you can get, in one sort of eye drop bottle sized mercuric chloride preservative, you can preserve hundreds, if not 1,000 samples, and you're dealing with small volumes in the field. Alcohol and formaldehyde are the quantities in which you have to add those will change the carbonate chemistry so significantly that those samples are probably not valuable for ocean acidification. Formaldehyde in particular in the formalin form is strongly buffered. And so it will change the acid-base chemistry if you add that to your sample. So the mercuric chloride, and particularly with these ion-specific filters that we're using, you can capture most of that mercury from your samples when you dispose of them, as opposed to returning it to the natural environment.
Burke, also maybe talk about there's no need to put it on ice, and then how long they're stable for.
Yeah, so once a sample is properly preserved with mercuric chloride, we think they're stable for months, if not years. And it's very important for us to know the temperature at which you collected the sample so that we can tell you about in situ conditions. But it does not have to be maintained at that temperature or kept cold once the sample is properly preserved and sealed.
Thank you.
All right, I think that was a really good question. And if there's one more question, I think we have time for one more question.
Sorry, Alexis.
Yes.
Sorry, this is Falilu. Actually, I was in transit when I sent in my comments, and somehow I lost connection, and I just got back into the room now. I just joined back the meeting. I just want to ask something very closely to what was answered, just concerning the mercury chloride. I'm sorry to take you back. I just want to know, what's the shelf life of mercury chloride solution? What's the shelf life, please?
So the way we prefer to prepare mercuric chloride is to make what we call a saturated solution. And so you take distilled water and you add mercuric chloride until it stops dissolving, and your stock reservoir mercuric chloride should always have the white solid precipitate on the bottom. When you go into the field, Again, we prefer to take the supernatant, the liquid solution from the top of that, and only take small volumes. A few milliliters will allow you to preserve hundreds of samples. And then you're not bringing the solid mercury into the field, you're not bringing large volumes, and the dropper bottle approach involves less human handling of the sampling interface. And we don't really see any evidence that mercuric chloride in distilled water solution has got a shelf life that impacts its preservative capability.
And Valauri, I did mute you just because there was some background noise, but
sorry, I'm sorry. Okay, then what about the temperature? Because what I'm saying is that when you have a mercury solution for your sampling, when you prepare one for now and you have to sample the other time, you might not want to discard, considering the environmental implication of discarding mercury chloride. But what about the temperature at which you can leave it in the sample container, the reagent bottle? What temperature can you leave it?
Once you've collected and preserved and sealed the sample, it's no longer sensitive to temperature in ways that we don't understand very, very well. So if you tell us with reasonable accuracy what the temperature of your sample collection condition was, we always measure in the laboratory that analysis temperature, and we know very well how to make the corrections from that analysis temperature back to your in situ condition.
yeah, and actually most of the ways that we do these analyses, that there's spreadsheets that do that calculation for you. If you input what the field temperature was and what the lab temperature is, it'll do that correction for you. So there's some really great Excel spreadsheets. You can also use MATLAB and other
Does
softwares.
everyone have, did we send out Dixon's guide to best practices?
I'm gonna send it out. We linked to it on Wednesday, but yeah, there's this very robust document that Andrew authored, I think with Chris as maybe co-author, I can't remember, that is just like a best practices, and it walks through a ton of this information. So we will.
It talks about like how to do quality control and how many duplicates to take per however many samples you're taking, and goes through a lot of the stuff that we went through today about each parameter. It's really useful guide.
Yeah, so I think that's the end of our time today, but we're just really happy that you all joined us, and I'm very appreciative for Sophie and Burke's time and Andrew joining us by phone to provide some of their expertise. But this isn't the end of our help. We, like I said, invite you to apply for a coaching session, and please email me and I'll give you other ways to connect to this network. And I'll always let people know if there's funding opportunities for equipment and other things. But at the very least, I can promise that there's the peer-to-peer scholarship, which we're able to fund every year. And hopefully there'll be more opportunities, but I'm not 100% sure yet because I'm waiting for some funding to come in. So thank you all so much. And I'll make sure the recording is sent out to everyone.
Thanks everybody for attending.
Thank you.
Thanks for coming.
Thanks.
Thank you. Bye-bye.
Bye.
Thanks, Alexis. Thanks, Sophie.
Alex and Sophie. And Hales.